Hello, everyone. My name is Linda Grillová, and first of all, I would like to say that I'm really happy that I have been invited to this forum. I have been working with syphilis for many years now, and currently I am a curator of PAP MLST of Treponema pallidum, subspecies pallidum. So today it's a great pleasure of mine to show you some of the results that I have been working on in the last few years in regard to MLST of syphilis. So in the beginning, I would like to give you a brief introduction about T. pallidum and about syphilis. Then I would like to talk about the design of MLST, how the T. pallidum MLST actually is different compared to other MLST in other bacteria, and how and why. Then in the end, I would like to share with you some of the discoveries that we made based on MLST. Many people think that syphilis is actually a historical disease. It's not true. If you can see in this map, the incidence is quite high. There is 5.6 million new cases every year, and the prevalence is still increasing. If you take T. pallidum and you compare it with other bacteria, you could see several unique characteristics. It's its unique spiral shape and corkscrew movement that enables T. pallidum to survive in high viscosity environment. And then that actually means that T. pallidum can actually disseminate to any organ in the human body that you can think of. It has extremely small genome, and this is actually the reflection of the fact that there are many metabolic pathways absent, and that T. pallidum is not able to synthesize nucleotides and fatty acids in a well. All these things actually can say that T. pallidum is extremely adapted to the human host, and this was actually the reason why we were not able to cultivate this pathogen in vitro until 2018. So before 2018, most of the experimental data came actually from treponems that were propagated in experimental animals, in experimental rabbits. After 2018, we are using in vitro cultivation in co-culture system. That means in the presence of other eukaryotic cells. But this system still requires state-of-art skills, and not so many people are able to do it. So to my knowledge, only 4 laboratories in the world are actually able to cultivate T. pallidum in vitro now. So it's still a little bit difficult. So you can imagine that most of the people are actually focusing on genomics, even though genomics was also neglected for many years because we were able to sequence genomes only from treponems that were propagated in rabbits. And in 2016, there were many enrichment methods introduced. They were based on fishing of either treponema cells or treponema DNA directly from clinical samples. And this was really good because it enables us to sequence genomes directly from clinical samples for the first time. So you can also imagine that molecular typing is a really powerful tool in this research. It has been, and it still is. Currently, there are 2 typing schemes, CDC typing, that have been introduced in the year 2000, and which is based on restriction fragment length polymorphism of different genes, and on the number of repetition in ARP genes. And there is multilocus sequence typing, that was actually based on several genomes that are sequenced directly from clinical samples, and that have been introduced only in 2018, because before we didn't have the genetic information that would actually reflect the real treponems that are circulating in the real human population. So in 2019, the MLST was public and was connected with PAP MLST. And since then, we collected around 1,000 isolate from different parts of the world. So we are very happy. But the truth is that most of the people are still using CDC typing. And I hope that in the future, many people will switch to MLST. While we were designing MLST, we were really careful which loci are we going to choose, because we wanted to have this kind of loci that have the highest discrimination power. That means that it has a high resolution compared to genome-wide data. But at the same time, we wanted to have this kind of loci that have a typing stability and that can actually give us some information about phylogeny. Typically, MLST is based on 7 housekeeping gene. T. pallidum MLST is based on 3 variable genes. And the reason is that the average nucleotide identity of T. pallidum is 99.8%. So T. pallidum is extremely monomorphic organism compared to, for example, E. coli, where average nucleotide identity is 95%. So this is really on the border of bacterial species. So if we would choose housekeeping gene in T. pallidum, we would end up only with 1 sequencing type. So this was the reason why we decided to go for a little bit more variable loci. At the same time, we wanted the loci that are actually able to somehow tell us something about phylogeny. So this is maximum likelihood phylogenetic trees based on recently sequenced genome. It was actually published by my colleague Matthew recently. It represents the biggest genomic data set that exists in treponems, and so you can see that it's absolutely obvious that there are 2 genetic groups, the blue one and the red one. We call them Nichols-like and SS14-like, and we can see the SS14-like are in red and the Nichols-like are in blue. You can see that there are much more SS14-like isolate compared to Nichols-like, and they seems to be quite clonal, and Nichols-like isolate seems to be a little bit more diverse. So this was something that we wanted to see also in MLST. So we choose 2 variable loci, 136 and 548, that were able to distinguish between SS14 and between Nichols-like strains, as you can see here. And then we also choose 705 locus that is not able to distinguish between Nichols-like and SS14-like strain, but is able to reveal several sublineages within SS14-like strains. So that was really important for us. In the end, the concatenator of these three genes actually gave us 40% of resolution of that have been previously achieved by genome-wide data. So I think that was pretty good. We also recommend to use or to sequence 23S just to check the presence or absence of point mutation that can lead to macrolide resistance. So actually, in the end, the MLST loci is based on 4 genes. And so since 2018, that MLST was developed, we have actually described population structure, and we have described the dynamic of treponema infection in different countries, in Europe, in America, in Asia. Sometimes we were able to enhance the diagnostic method. For example, in Cuba, we figured that some of the patients that were actually diagnosed with venereal syphilis with other diagnostics tools, for example, with serology, were actually patients that were not infected by Treponema pallidum subspecies pallidum, but with subspecies endemicum, which is causing different disease. Then we found several associations between allelic profile or sequencing type, clonal complexes, and some metadata of the patient. So we noticed that Nichols-like strains are actually more prevalent in men who have sex with men, in homosexuals, are more prevalent in a little bit older population, and we also noticed that most of the SS14 strain actually carry point mutation for macrolide resistance compared to Nichols-like strains. And in general, we got this view about the distribution of SS14-like strains and Nichols-like strains. And so as you can see here, SS14-like strains are in yellow and Nichols-like strains are in red. So you can see that SS14-like strains are really predominant in most of the countries that the analysis have been done. And this is really something interesting, because Nichols-like strains are well-studied strains compared to SS14-like strains, because they have been passed through rabbits, and they were growing in rabbits historically very well compared to SS14. So some of the people think that SS14 actually have some biological advantage in regard to human infection, and the other way around, that Nichols-like strains have some biological advantage in regard to rabbit infection. Some other people think that it's just sequencing biased. Some other people think that actually Nichols-like strain are determined to be extinct and replaced by SS14-like strain, because they are simply just older. So this is something that definitely has to be still addressed. But for this, we definitely need to have a little bit more information and continue working with MLST. So I would like to thank all the people that have been involved in MLST design. And also I would like to thank people that decided to use MLST. And in the end, I would like to thank you for your attention. And I think that I will be ready for your question.